Hello, everyone. Good afternoon. My name is In, so I'm the product manager from Crystal Bio. It's my pleasure to have all of you in our webinar today. Today, we will talk about the troubleshooting and guidance for working with ELISA. And for today's webinar, there will be two parts. The first part will be the interpretation of the ELISA test result. And the second part will be like when you have the abnormal result during the experiment, how can we analyze it? And then how can we improve our experiment and to make the next ELISA experiment to be good? So let's start the journey today. The first part, I'm going to talk you like how we can make the judgment from our data. And then basically they can come from 4 different aspects. So the first one that I showed you is the color development. So as you see, the color development is a pretty straightforward visualization of the color change with our naked eyes. And in the ELISA, we usually have the standard proteins, right? And based on the standard proteins, we can build the standard curve. And in the color development, it's kind of like in reaction between the substrate and the enzymes. So for example, here in this assay, the TMB is the substrate for the HRP. So before we add the stop solution, the color is usually blue. And then after the addition of the stop solution, the color will turn to yellow. So with the standards, we can distinguish whether there's a clear gradient color with the naked eyes. So if you see this clear gradient color, then it's highly possible that the ELISA experiment is pretty good. So this is the first aspect. Then for the next aspect, we also know that after the color development, we will move our plate into the plate radar. So we can tell from the OD values to make a judgment. So for the OD value here, I also have one example from the standard. So you see that we can check the high concentration group. So you see the OD is greater than 1.0. And for the low concentration group, the OD is less than 0.2. And if for the example 2, we also have 3 samples. You see that the OD values for the samples, they actually fall within the standard range, meaning that if you see the values here, they actually vary from, they are kind of like between the 0.17 to 2.5. So with the OD values, you can also tell whether the experiment is good or not. So then the next aspect is the correlation coefficient. It's also related to the standard curve because the correlation coefficient is a value can express the precision of the linearity fit of the experiment data. After we got the data from the standard curve and using the software, it's easy to get a linear regression. And with the linear regression, we can get the value called R, and it's the correlation coefficient. It's kind of like it can determine the overall goodness of the fit. So I will say that if the R is larger than 0.99 to fit, and the data looks fine. The last aspect I will say is the accuracy. For the accuracy is commonly represented by the coefficient of variation. So let's say it's CV. With the CV, the calculation is quite based on the standard deviation and the average value between the 2 replicates. So the CV can help us to identify any inconsistency and inaccuracy in the result. So the larger the variance, the greater the inconsistent and error can happen with the experiment. So usually for the standards, we require the CV to less than 8%. And for the samples, we expect the CV to less than 20%. So here's one example from our Crystal Bio ELISA kit. You see, we have 2 replicates for the standard, OD1 and OD2. Based on the average and the standard deviation, we calculate the CV. The CV overall is less than 8%. So this set of data is pretty good and excellent. And meaning that at least the standards, the ELISA experiment is good. So we can move on to the next to analyze our samples. So all in all, with all these 4 aspects, we can use them to determine whether an experiment is good enough and help us to perform the following analysis. So after I talk about the second part, I always know that the results, sometimes they are not that ideal in the reality. And then the analysis for the abnormal result will be very important to help us to, to get the next one to be good. So here is a summary about the 4 common problems that people may find in their ELISA experiment in the reality. The problem 1 is that what about like my whole plate is with very high signal. So meaning that there's high background there or non-specific binding there. And then the problem 2 is the opposite. So what about there's no color or light color development, including my standards, so across the whole plate. And the problem 3 is that the standards look fine, but why the signal in my samples, they are pretty low? What happened to it? And the last problem is that about the reproducibilities. So, the whole plate, we have a lot of standards and the samples. Sometimes there will be unexpected result shows up, making the data looks that not a little bit odd. So how can we deal with it and what the possible causes behind it? So for all these 4 common problems, I will explain them one by one to tell you like the possible causes behind these problems and what solutions can we take to solve them. So let's start the problem with the problem 1. As you see here, I have a figure here from the real experiment. You can tell the color about the standards. So the first line is the color development from the standard. You see all yellow was observed across the plate. And even with the low concentration group, we expect to see a clear color because they are with the low concentration, but actually they are not. They are just all yellow. And the same with the OD values. So you can tell from the OD values that at the high concentration group, the gradient of the OD rating is well. However, in the low concentration group, most of the ratings, they are over 0.3, and there's no clear gradient observed. So for the example number 2 here is the same phenomenon. So with this, we will say that the whole plate, they probably have non-specific binding, causing this high background phenomenon. To solve this problem, the first thing I would suggest or highly recommend it is to check all your reagent and the buffers, especially like, we usually bought the ELISA kit. Then did the ELISA kit expired? And what is the lot number of the ELISA kit? And are all these reagents, they come from the same kit? So if this is the case, please be sure to, check all of them. And if the buffer's getting old, then you can prepare the fresh buffers with the fresh filtered water. And then the next possible cause can come from the insufficient washing. Because insufficient washing, they usually cause the non-specific binding remaining in the plate. So especially like some unbound antibodies. And these unbound antibodies, they will kind of like, causing the non-specific binding. So for the wash, I have a small suggestion here. When we do the washing, we can invert the plate on the absorbent tissue and type the plate to remove any residue fluid and allow the plate to completely drain. So with this, and have the consistent washing step during the whole ELISA experiment, you can have enough washing to get rid of the noises. And the next 2 is about the longer incubation time or longer waiting after you add the stop solution. For the longer incubation time, is usually can affect the binding process between the antigen and the antibody. And this will also lead to a non-specific binding, increase the cross-reactivity, right? And for the next one, so a long color development time, it also can affect the enzymatic reaction because the enzymatic reaction usually happens between the substrate and enzyme. So too long reaction time will result in the increased background signal. So inaccurate final ratings will get. So all this, they will, affect the final rating. So some other possible cause in the problem 1, I will say, is related to the excess concentration, the high antibody concentration. So think about the excess antibody. Not all of the antibody, they will bind specifically to the target antigen, and especially the unbound antibody. They can adhere non-specifically to the plastic surface of the microplate wells. So this unspecific binding will result in a strong color development reaction. So, and I also want to remind our audience about two concepts, the stop concentration and working concentration. So I know that when the ELISA kit come, the kit, they always have the buffer with the stop concentration. So then you need to dilute the stop concentration to get an optimal concentration called working concentration. So it's different. You need to use the buffer with the working concentration for your ELISA experiment. And some other relevant cause that I would say is the HRP conjugate substrate contamination. So no matter this contamination comes from the plates or the reagent reservoirs or the pipette tips. So be sure to avoid this contamination. And the best solution to avoid it is to use the flash plate Sarah or the new reagent reservoir. Also, be sure to change the pipette tip when you do the pipetting, especially when you pipetting different reagent. And also another thing about the substrate, I will say, is the sensitivity. So substrate, especially the chromogenetic and the phylogenetic substrate, they are pretty sensitive to the light. Exposure to the light can cause them to undergo some unwanted chemical reaction. So people may ask, how can I tell that whether my substrate solution goes bad? Let me give you a tip here. So you can tell from the color change of the substrate solution. Usually the substrate solution is clear or slightly blue, but when you see the color from your substrate solution, if the color already turns to blue, then my recommendation is to replace them or prepare a fresh substrate solution for your ELISA experiment. And also another thing I want to remind you is that sometimes after the color development, you will use your naked eyes to see the color, right? And then you saw that the color gradient looks good, but then when you move to the plate reader, the rating values are pretty odd and it's kind of like unexpected. Then for this case, I will suggest you to check your wavelength of the plate reader. So which means that for some specific substrate, you need to use the corresponding wavelength. So for example, for the TMB substrate, the primary wavelength is typically 450. And so be sure to use it for your TMB substrate. And another thing, like, I think most of you probably wondering it's hard to distinguish which type of the wavelength. Then for this case, my suggestion is to ask your kit supplier. I think you will get the answer from them. Yeah. After I talk about the problem one, the problem two here is the opposite with the problem one. So instead of high background and high signal, what happens if my plate is no color and light color? So here's one example about this situation. So take a look at the OD here. This is the OD from the standard, and you see that all the ratings, they are below 0.1, and there's no gradient here. And with our naked eyes, the color of the whole plate actually is pretty clear. You know, it looks like a very, very clean plate. And for the weak signal, so something like this. So this is the standard. You see the color, but the color is really significant weak. And for the OD values, you check the OD values, they are also pretty low. Even with the high concentration rating, the rating is slightly above 0.5. So then what are the possible causes then causing this no color or light color? Common reasons that I will say they are the mix of the reagent, or you missed some key reagent or key step during the ELISA experiment. For this case, I will say that check all the reagent and then follow the instructions strictly to finish your experiment. And moreover, some possible causes, they can be from the color development step. Like if the color development goes wrong, the color development, then you will never get any clear color. So as in the ELISA, the last step is always the reaction between the enzyme and the substrate. So we need to really be careful about this part. And this part, They usually because the substrate is no longer the HRP conjugate is no longer active or too old, or the whole kit it just expired, and also maybe because the storage is not that good, so the kit goes bad. So for this, you can check the expiration date, and or even after you get the kit, make sure the kit is stored in a very optimal condition to keep it safe. And another tip is that you can also test the conjugate and the substrate for the activity, or just use the fresh reagent if you are worried about this situation. And another situation pretty similar to this one is about the enzyme inhibitor. It's also related to the substrate. So if the reagent, they have the enzyme inhibitor, such as the sodium azide, this kind, they will kind of like interfering with or completely block the enzyme activity. So then affecting the color development reaction, resulting in the diminished signal that you saw. So be sure that your reagent are prepared good and they are prepared with the qualified distilled water. Similar with the first problem, there are also a lot of general costs that need to be considered. For example, the assay temperature, the incubation time, and the color reaction time, and also even the concentration of your detection reagent. So all this, they will also affect your result to get the no color or light color. So for example, one tip here, before you start your experiment, you can bring all your reagent to the room temperature for 30 minutes before you use it, but be sure do not leave it in room temperature for a long time. And other is that usually with the kit, the manufacturer will give you like a proper guidelines about the, how long is the incubation time and how long is the color development time. So just be sure to follow them to get your experiment work. Another some of the possible causes that I hear is not that common, but I still want to remind you. So for example, some of specific kits, they have two different substrates. So for these two substrates, let's call it substrate A and substrate B. It's common to add the A first and the B later. But if you add the A and B in a correct order, then the experiment will not work. So for this potential cause, just review the instruction and repeat the assay again to make it work. The last set of the possible reasons that I will talk with the problem 2 is about the operation errors that people may have when they perform the experiment, such as the pipetting techniques and the washing times. So for the pipetting techniques, so sometimes it's not just the technique, it may be because the pipette itself. So my recommendation is that please calibrate the pipette regularly, especially like once a year. And also when you performing the pipetting, make sure that the tips, they fit tightly with the pipette. Also for the washing times, my suggestion is that accurately record the number of the washes, and then keep an eye on the volume of the wash buffer that you used, and strictly follow the instruction, dilute the wash buffer. As I mentioned before, sometimes the wash buffer, they have the concentrated concentration, not the working concentration. So be sure to distinguish these two types of concentration. So after I talk about the problem 2, let's move on to the problem 3. Problem 3 is different from the previous 2. So in this problem, usually the standard all looks good, but it's just the samples. The samples, they are themselves, they have a light color here. So one example that I showed here. So this is the interleukin-2 ELISA kit from our Crucible, and we have the standards here. So take a look at the color development and also the OD values. All of them, they looks good. There's a very clear gradient here, and the OD, they have a very good value range. But when we come to the samples, you see like the color are pretty clear, and also the OD values are quite below the suggested low concentration group. So is a little bit odd, right? So for this problem, I will say that most of the possible causes, they come from the samples. So the first definitely will be, did you check your sample preparation? And did you check the storage of your samples? Are they good? If that is the case, then the best solution will always be like prepare your sample again. And when you store your samples, try to avoid contamination. And the next possible cause is about the target concentration. So as I showed you before, in the Bio ELISA kit, the kit is about interleukin-2. And we know that the interleukin-2 from the human serums, they always have a very low concentration. So for this low concentration samples, sometimes the concentration, they are below the minimal detection limit of that kit. So if that is the case, you can either concentrate the sample or you can reduce the sample dilution if you performed one. But I think another better solution is to contact with the supplier to ask them whether they will have a kit with the higher sensitivity for your samples. You know, replace your current kit, get a more sensitive one, and then repeat the experiment again. The third possible cause about the sample, about this problem, is calling the hook effect. Hook effect is pretty common in the ELISA experiment. So this is that your sample, it has a very high concentration, but because the dilution factor is too small, so it can lead to a hook effect. Here's a diagram showing that how the hook effect can happen. So usually like in the sandwich ELISA, the capture antibody is immobilized on the solid phase, and the analyte can be captured with the captured antibody. So then after you add the substrate, and will result in a color change and the signal will increase. However, if you have a very high analyte concentration, the excess analyte can saturate the capture antibody here. So there will be a lot of free analytes in the assay solutions. And this free analyte solution, they compete with the capture antibody and blocking or reduce the effective binding between the detection antibody with the analyte. So resulting the signal increase a lot, then decrease. So this is a falsely low signal that you may observe. So this is the reason from this is called the hook effect. So with this, the suggestion is to increase the dilution factor of the sample to avoid the hook effect. Next, the two possible reasons, they are also related with the sample. One thing I want to remind you is about the matrix interference. The matrix interference, they come from the extra elements in the samples. So in the samples, they may have the proteins or the lipids, and they may disrupt the binding between the target analyte and antibody. So the solution for the matrix interference is you can perform appropriate dilution to decrease that noisy. And also you can use like a positive control in your assay to help you to get the real data. The last situation is about the sample type. We know that sometimes we not just detect the analyte, but sometimes the sample can be the recombinant protein. For the recombinant protein, they are different from their nature counterpart in the structure or the post-translational modification. So for this type of the analyte, our suggestion is to get suitable antibody pairs for this recombinant protein. And also, if you are unsure about which kit you can use, always contact with the kit supplier for advice if you are unsure about it. So the last problem from the ELISA experiment is about the unexpected result and the poor reproducibilities. So here's one example. We have the standards and OD1 and OD2. You see, they are so different. And because it's different, the CV is around 31%. It's definitely higher than what we required. And the color, and also may sometimes due to the unexpected result here, causing there's no clear gradient. So the result looks odd. And another example here, the CV also looks pretty, that optimal. So we are worried about this result, but how can we solve them? Possible reasons, I will say, is that first, think about your standard solution, especially if you find the unexpected result in your standard solution, because usually the kit, they comes with the standards, and the standards, they are usually the lyophilized standards. This kind of standard, they require the solution with the recommended diluent. So be sure that your standards, the dilution is good, the preparation is good. Then make sure that at least in your experiment, the standard curve is good. Then the next I will say is recommended to consider all the improper operation during the ELISA experiment. Like for example, did you mix your reagent completely? And do you have a cross-contamination there? And also, did you do enough washing? And did you have a pipette error? So all this, I will call them improper operation. And one thing that about the cross-contamination is about the washing. So I think I tell you that we usually tap the plate generally on the absorbent paper to move the excess wash buffer. But when you do the washing, try to avoid the splashing between the wells. And also, don't forget to change the pipette tip to avoid contamination. So all this, they are pretty easy, pretty important. And also ensure the consistency across all the wells when you do the washing. For the pipette, and I think I said a lot, please be sure about your pipette has been calibrated regularly. And also, another suggestion about the pipetting technique is that you can do the practicing of the pipette technique using the water, multiple times before you start the ELISA experiment. So some causes, they can come from the environment, like the incubation environment. But here, I want to remind you another thing, the edge effect. So we usually performed our ELISA experiment with the 96 well plate, right? Or the microplate. The edge wells, they are so easily affected by the variable environment, like the temperature, the humidity. So this can cause the evaporation rate be different between the wells. So then the solution for this kind of a possible cause is to use the edge wells for the negative controls, or you can use the sealing films. And also be sure that when you do the incubation all the time, don't let the plate close to any heat source to affect the evaporation. And also the assay setup. So one tip that I said is that be sure to bring all your reagent from the fridge to the room temperature 30 minutes before you start the ELISA. But do not leave all the components at the room temperature for too long. It will definitely affect the final ratings. The last two I mentioned here in this slide is about the transmission. So after the color development, we move our plate into the spectrophotometry. And we know that for that plate radar, the light, they usually comes from the bottom of the wells. So the scratch or the dirt on the bottom of the plate or on the wells and the bubbles, all these factors, they will block or scatter the light. So this situation will lead to the inaccurate measurement. So be sure that when you do the pipetting, do not touch the bottom of the plate. And when you see the dirt or the fingerprint, just quickly wipe the bottom of the plate to avoid it, to get rid of it. And also for this problem, there are some external factors, especially the causes that can come from the samples. One thing about the sample is that check the storage of the samples. Are they too old? if they are too old, my suggestion is to make fresh samples. And another thing about the sample, after you take them out from the fridge, is make sure that you centrifuge them before you add them to the ELISA plate. Because we know that the clotting can happen due to the interference caused by the residue serial component. So they settle down, then they will block the light, and then they will affect the final reading. Last possible cause, I think this one is related with the qualitative kit. With the qualitative kit, usually when we make the decision, we just make that whether is it positive or negative, right? But sometimes the signal intensity fluctuation, they will affect the decision because there's usually some small variation that fluctuate between around the threshold. So thinking about you sometimes get positive, you sometimes get negative. So for this possible cause, I will say that you can conduct the experiment multiple times and independently. Then, like for example, you perform the experiment 3 times independently, and then 2 of the results are positive and then 1 is negative. Then it's highly possible that it may be positive, but I'm just saying highly possible. I'm not saying that it's definitely right. But another better option I will suggest is to set up the positive and the negative controls to ensure the reliability and the accuracy of the result. And with this, you can quickly make a decision from there. So overall, at the very end, I will say that when you get the abnormal ELISA result, don't worry. They come from so many sources of the interference. So regarding that, here I just introduce our quickly workflow to guide you from the sample preparation and to guide you to analyze the interference from outside or from inside before you make the analysis. You can quickly just follow this pipeline to exclude any possible causes that may happen. And also here, this is a brief summary of the interference in the ELISA experiment, like some incubation conditions. Did you check them? Did you check the place setting? Did you check your kit? Are they expired? Are they stored good? And also, did you check the concentration of your conjugated enzyme? And also, did you care about did you take a look at your samples? Are they too old? Did you think about the interference from the sample matrix? Did your samples that diluted good? There's so many interference here. And I really hope that with this brief summary, you will get your experiment going good. Yeah. So then at the very end, please give me several minutes to introduce our company. We are Crystal Bio. Crystal Bio comes from a customer-satisfied biology. And except the ELISA kit, we also have a different product lines. For example, we have the proteins, we have the antibodies, we also have the molecular biology products and the small molecular antigens. Our products has been exported to more than 80 countries, including the Europe, American, Japan, and Germany. And as you see, and also we have a very wide cooperation with famous companies like Pfizer, Sanofi, Zinker, Johnson & Johnson. So, our products are pretty popular. And at the very end, thank you so much. If you want to stay connected with us, please follow us in the LinkedIn, and you can communicate with us, and you can ask us questions, and whatever you want. Thank you so much, and I will take any questions.